Hello guys, today we are going to start this chapter which is body fluid and circulation. This is from unit Human Physiology of class 11th NCERT. So let's begin. So we all know that every living organism, all the living cells have to be provided with nutrient, oxygen and essential substances, and waste, harmful substances have to be removed continuously for healthy functioning. If toxic waste or harmful substances are accumulated inside our body, then they will cause a lot of problems, a lot of disease in our body, right? So basically it's essential, very essential for us to have efficient mechanism for movement of these substances from cells and to the cells. So the different groups of organisms, they have evolved different methods for transportation. For example, simple organisms like sponges and cylindrids, they circulate water from their surrounding through their body cavities to facilitate their cells to exchange these substances or for transportation. So they use water from the surrounding for transportation and exchange, in their bodies. And more complex organism, like in case of humans, blood is the most commonly used body fluid, or which is used in larger organism, higher organisms like humans. Lymph is basically another body fluid which helps in transport of certain other substances. In this chapter we are going to learn about blood, lymph and mechanism of circulation of blood. All of that will be explained here. Now, blood is basically specialized connective tissue which consists of fluid matrix, plasma and formed elements, but no fibers. Okay, so plasma is basically a straw shape which is yellowish color, viscous fluid which basically it constitutes 55% of the blood is plasma. 92% of the plasma is water and protein is about 6 to 8%. Now major proteins in plasma are fibrinogen, globulins and albumins. Alright. Now we have fibrinogen which is needed for clotting or coagulation, globulin which helps in defense mechanism of the body, and albumins which help in osmotic balance. These are the major proteins present in plasma. And basically in plasma 90 to 92% is water and 6 to 8% is protein. And in blood we have 55% plasma and 45% of blood is formed elements. Now talking about more components of plasma, so plasma also contains small amount of minerals like sodium, calcium, magnesium, bicarbonate, chloride, glucose, amino acid, lipids are always also present in the plasma, and they always transit in the body. Okay, now factors for coagulation or clotting of the blood is also present in plasma in inactive form. Of course, fibrinogen is the protein which is present for clotting or coagulation, but I'm just saying that factors which are also responsible and they're in inactive form and responsible for coagulation, clotting of blood, they're also present in plasma. And if plasma is collected without clotting factor, then it's called serum. Means plasma without clotting factors is called serum. We have formed elements. Formed elements like erythrocyte, leukocyte and platelets, they constitute 45% of the blood. Means 45% of the blood is formed elements. Now erythrocyte, this paragraph is for erythrocytes, which means red blood cells, RBCs, most abundant of all cells in our body. Their number is 5 to 5.5 million of RBCs are present per millimeter cube of blood. They are formed in red bone marrow, devoid of nucleus, biconcave in shape. They have red colored iron pigment and its complex protein which is called hemoglobin. In 100 ml of blood, we have 12 to 16 gram of hemoglobin on an average, or we would say that healthy individual has that. Now, these molecules which are hemoglobins, and what are these? Red colored iron complex protein. And these molecules have significant role in transport of respiratory gas, which means hemoglobin help in transport of respiratory gases, and hemoglobin are present in RBC, so we can say RBC help in transport of respiratory gases. RBC have lifespan of 120 days and they are destroyed in spleen, which is also called graveyard of RBCs. Then we have this paragraph for leukocytes, which are WBCs, and they are colorless because of lack of any pigment like hemoglobin. They are nucleated. However, red blood cells were enucleated, and on an average 6,000 to 8,000 per millimeter cube of blood WBCs are present. And then we have, they are short-lived. And we have two main categories of WBCs, which are granulocyte A, granulocyte. Granulocyte consists of neutrophil, basophil and eosinophil, and agranulocyte consists of monocyte, lymphocytes. Also, let me tell you one thing, neutrophil is 60 to 65% of total WBCs are neutrophil, 0.5 to 1 0.5 to 1% of WBC is basophil, 2 to 3% eosinophil, 6 to 8% monocytes, and 20 to 25% lymphocytes. The largest or most abundant granulocyte or WBC is neutrophil, and least abundant granulocyte or WBC is basophil. This is a sequence you can remember. Neutrocyte maximum, then we have lymphocyte, then we have monocyte, eosinophil, and then basophil. You can remember their shape that is given here in the photo. So basically they can be asked by showing you the diagram and asking you that which of them is which one, right? Which WBC is this? So they can ask questions like this, is headphone shaped nucleated eosinophil. Then we have basophil like this. Then we have neutrophil, which is which has multi-lobe nucleus, and then bilobe nucleus present in monocyte. T lymphocyte and B lymphocytes are also there. Lymphocyte types is basically T lymphocyte and B lymphocytes. Now we have neutrophils, 60 to 65%, basophil 0.5 to 1%. Neutrophil and monocytes both are phagocytic in nature and destroy foreign organisms entering the body. And then basophils secrete histamine, serotonin and heparin, and they're involved in inflammatory reactions. Eosinophil resist infection and are also associated in allergic reactions. Then we have lymphocytes. They are of two types, and both of them are responsible for immune responses in the body. Megakaryocytes, basically platelets, are formed of megakaryocytes. Basically platelets are small fragments. These are formed from megakaryocytes, which are present in bone marrow. Alright, now we have the number of platelets per millimeter cube of blood is 1.5 to 3.5 lakh. Then platelets are important, or they play an important role in coagulation and clotting of blood. We'll talk about coagulation, clotting of blood in later half. But first of all, reduction in the number of platelets can cause severe damage to the body, because it can lead to clotting disorder, and that will lead to excessive loss of blood from the body. Now talking about blood groups, we have two types of blood groups, ABO blood group and RH blood grouping. So ABO blood grouping is based on presence absence of two surface antigens which are present on the surface of RBCs. And what are the antigens? These are chemicals that can induce immune responses. Like for example, if a antigen is present, then it can induce immune responses. That's what I'm trying to say. And so two surface antigens are A and B. Plasma of different individuals have two natural antibodies, which are proteins produced in response to antigens. So proteins produced in response to antigens are called antibodies, and plasma has two natural antibodies, that is anti-A and anti-B antibody. And if we talk about the types of blood groups, we have A, B, AB and O. On what basis we'll talk about later, but first of all you have to know that blood group of donor or blood of donor has to be carefully matched with the blood of recipient before any transfusion of blood to avoid severe clumping or destruction of RBC. So you have to remember one point, that the antigen of the donor should match with the antigen of recipient. If there is any mismatch of antigen of donor with the recipient, then the recipient RBC is going to destroy, or there would be destruction of RBC or clumping of RBC in case of a recipient, and that would be severe, and that would be lethal. Now blood group AB and O have antigens on RBCs. A has A antigen, B has B antigen, AB has both these antigens, and O has no antigens. A blood group has anti-B antibody. Why? Because if it would have A antibody, anti-A antibody, then it would kill its own antigen, right? Because antibodies are what? They kill antigens. That's it. So for A blood group, we need donors who have A antigen only. let's say who have A antigen or who have no antigen, because we all know that antigen of the blood donor should match with antigen of the recipient, because antigen is more important because they are present on the surface of RBC. However, antibodies, if you talk about why antibodies should not be matched, or why should not consider that, because there are certain things that come in mind, the doubts. So basically antibodies can be diluted because they are present in plasma, so they can be diluted. However, if we talk about antigen, they're present on RBC, they're going to carry it through the blood, they're the cells, they cannot be removed, right? So the antigen has to be taken in care of. So donor's antigen should match with the recipient's antigen, and simply so we should consider that in mind before transfusion of blood. Now for A, it has A antigen, A blood donor also has A antigen, O blood donor has no antigen, so it can easily give it. Now if we talk about B blood group, has anti-A antibody. Why? Because anti-A antibody cannot destroy antigen B, right? And donor B has antigen B and donor O has no antigen, so both of them can be given to the blood group B. Now we have for AB, it has all antigens, so all the blood groups can be given, because there are only two types of antigen, no? So all the blood groups can be given, because there would be no mismatch. Even if, for example, let's say B blood group is given, B blood donor has B antigen in it, and B antigen is matched with the B antigen present here also, right? So there is no mismatch, and therefore it can be donated. For O blood group, it has no antigen, so O blood group has to be donated by only O blood group. Why? Because if any of the blood group like A, B or AB, all those blood group would have certain kind of antigen, but it has no antigen, so there will be a mismatch, and the blood of the donor when come in contact with the blood of this O, like let's say A comes in contact with O, then A antigen from the donor will be killed by O antigen. How? Because O blood group person, because O blood group will generate anti-A antibody, because that's the foreign particle, right? Anti-A antibody will be generated in the body of O against the antigen given by this blood group A, right? So it will generate anti-A antibody, and this anti-A antibody will actually destroy its own RBCs, right? It destroy the RBCs given by the donor in the blood of recipient. So there will be destruction of RBC, there will be clumping, and it will be severe. Okay, it will be severe. So we have to carefully match the blood group of donor and recipient before transfusion. Now O is called universal donor and AB is called universal recipient. Why universal donor? Because it has no antigen. Okay, O has no antigen. Because it has no antigen, so you can understand very easily that it can give its blood to anyone, right? Because there will be no mismatch of the blood. And if we talk about AB blood group, then AB blood group has all the antigens, right? It has all A and B antigen. So in that case, it can only take blood, it cannot give blood to anyone, because all the antigen it has. So when it will give blood to anyone except itself, of course, like except the person who has AB blood group only, if it will give blood to A blood group, let's say, then the B antigen present in this AB blood group person will actually cause destruction of RBC in case of A blood group person, whoever it gives it to. So basically it can only take blood, cannot give blood. That's why it's called universal recipient. I'll talk about this also. This is about RH antigen and RH blood grouping, but O minus is universal donor and AB plus is universal recipient. Universal means that it can only donate, cannot take blood, and universal recipient means it can take blood from all and cannot donate. Of course, it can donate to its own blood group, it can also donate to its own blood group, but it can only take, it cannot donate, and it only donates, it cannot take. You understood? Now we have RH grouping. RH grouping is based on RH antigen. RH antigen is basically present in rhesus monkey, and this RH antigen is present in rhesus monkey and is also observed on the surface of RBC, a majority of humans, like 80% of humans, have this antigen on the surface of RBC. Now the individuals who have this are called Rh positive, and when Rh antigen is absent, then they are called Rh negative. 80% of the people have this, they are Rh positive. Now we have Rh negative person. When Rh negative person is exposed to Rh positive person, will form specific antibodies against Rh antigen. Like for example, I told you that if for example there is a recipient and it's exposed to B blood group, let's say, and this is donor, so a recipient is exposed to B, then the antigen, B antigen, basically against B antigen, A blood group will form anti-B antibody, and this anti-B antibody will kill the B antigen, and this, because this foreign particle, this foreign particle for this blood, so it will kill the B antigen, and there will be destruction of RBC and stuff. So that is what is written. If negative person, negative Rh antigen person is exposed to positive a person, which means Rh antigen is present in that blood, and that blood is basically exposed to a person where Rh antigen was not present, then in that case the Rh negative person will form specific antibodies that will kill this Rh positive person, or we would say that will kill the Rh antigens given by the donor to this recipient. And when it will kill, then there'll be anemia for that person, RBC will be destructed, it will be severe. Now Rh group should be matched before transfusion, that's also important. Now there is one special case of incompatibility, which is Rh incompatibility mismatching. What happens is that Rh negative pregnant woman, if that is there, with Rh positive blood of fetus, for example, if Rh negative blood is married with Rh positive blood, then there is a chance that the fetus can be Rh positive blood, right? So that is what I'm saying, that if Rh negative mother is there and Rh positive blood of fetus is there, then in that case Rh antigen of the fetus do not get exposed to Rh negative blood of the mother in first pregnancy. Why? Because placenta is there guiding, separating both of them. So placenta is basically separating the fetus and the mother, so fetus blood is not in the contact with the mother's blood. That's what is there. But however, during the delivery of the first child, possibility is there that mother blood gets exposed to the small amount of Rh positive blood from the fetus. Now what will happen during delivery, if mother blood is let's say is exposed to the fetus blood, which is Rh positive blood, and this is Rh negative blood, then in that case, due to this positive blood, Rh negative blood will start forming anti-Rh antigen, right? Anti-Rh antibody. Now this anti-Rh antibody, will actually cause problem, right? How problem? Because in subsequent pregnancies, in subsequent pregnancy, what will happen? Rh antibodies from the mother, which is Rh negative person, can leak into blood of the fetus, which is Rh positive, right? Rh positive has this Rh antigen, and the mother actually has antibodies against Rh antigen, and if it comes in contact with this in the subsequent pregnancy, then that will destroy the fetus RBC, and the fetus will have anemia after that, because the fetus RBCs are not destroyed, so there will be less RBCs for them, and there will be less transportation of oxygen because there will be less hemoglobin and stuff, so there will be anemia for that person, and similarly jaundice also to that baby. And because of anemia and jaundice, because of this condition, so this condition whole as a whole is called erythroblastosis foetalis, right? Now, but this condition can be avoided by administering anti-Rh antibody to the person immediately after delivery of the first child. So what is important to understand is that if we artificially give anti-Rh antibodies when the mother blood was exposed to the small amount of Rh blood in the first pregnancy delivery, then in that case, if we give it artificially, so I'm not able to write, but if we give it artificially, then in that case this artificial antibodies will actually get removed, or will actually get destroyed, or we would say that it will vanish from the blood. It will itself vanish from the blood, because it is artificially given. It is not going to persist. like the antibodies, if for example antibodies are formed in the mother's blood by its own body, then though it will persist and the mother blood will have this Rh antibodies, and when Rh positive fetus is again in subsequent pregnancy formed, then in that case, you know what will happen, this Rh positive fetus will be attacked by the antibodies of the Rh negative mother that were formed in the first pregnancy delivery, and that Rh antibody will actually destroy the RBC of the fetus, right, if it comes in contact with it. So basically what will happen is that in that matter it will have erythroblastosis fertilis. But if this Rh antibodies of the mother is given to the mother artificially, then it will of course destroy that Rh positive antigen given by the fetus when it was exposed. It will destroy that, and then it will vanish because it's not going to persist, it's chemically given. It will vanish, and because it will vanish, then in that case in subsequent pregnancy it will not cause problem, because there will be no Rh antibodies from the mother because it has already vanished, and because of that there will be no destruction of RBC of fetus, and there will be no erythroblastosis fertilis. I hope I made sense, right? Now in that case we can avoid this condition immediately if we give this negative Rh antibody. Now we have to talk about coagulation of blood. Now this coagulation of blood is important to understand because our body is capable of coagulation, which means that blood has this property of coagulating in response to any injury or trauma. This mechanism is to prevent excessive loss of blood from the body, and you all must have seen the dark scum that is formed, dark reddish brown scum that is formed at the site of cut from the injury over time. Now this is called a clot or coagulum, and this coagulum, what is coagulum? This is a network of threads, and threads are basically fibrin where damaged or dead formed elements of the blood are trapped. Alright, now basically this fibrin which was there in the scum, how these fibrins are formed? So we have to understand that these fibrins are formed by conversion of inactive fibrinogen by the enzyme thrombin. So thrombin is the enzyme which is present in the plasma. All these things are of course present in plasma. So thrombin is the enzyme which helps to convert fibrinogen into fibrin. Now you know this thrombin, how this thrombin is formed by inactive substance present in the plasma which is called prothrombin, and this prothrombin is formed by thrombokinase enzyme. you know prothrombin is converted to thrombin with the help of thrombokinase enzyme, and this thrombokinase enzyme is formed by series of enzymic reaction cascade process that are there involving the number of factors present in the plasma in inactive state. So basically all the enzymic reaction give rise to thrombokinase. This thrombokinase helps to convert prothrombin into thrombin, and this thrombin helps to convert fibrinogen into fibrin, and this fibrin is basically that was there in this scum, so it helps to form the clot and stuff. So now you can understand very easily with the help of this schematic diagram which I have drawn here. Is cascade process leads to the formation of thrombokinase, which actually converts prothrombin into thrombin, and this thrombin helps to convert fibrinogen into fibrin. This thrombin and thrombokinase are two enzymes that are there. Alright, now one more thing which is important to understand is that injury or trauma stimulates platelets to release certain factors that activate the process of coagulation. However, not only platelets can stimulate or can activate the mechanism of coagulation. Tissue can also release certain factors at the site of injury. Tissue can also release certain factors which can stimulate or which can activate the process of mechanism of coagulation. So they can also initiate the coagulation. So only platelets can initiate the coagulation. That's false statement. Both platelets and tissue at the site of injury can initiate the coagulation. Calcium in ions are also important for clotting, and we also know vitamin K is also important for clotting. Now let's talk about lymph. Now we know that lymph is tissue fluid. also known as tissue fluid. See, what happens is that as the blood passes through capillaries in the tissue, some water soluble substances move out in the spaces between the cells of the tissue. So what happens is that, let's say this is the capillary which comes in contact with the tissues, the cells in the tissue. So water from this capillary oozes out. Let's say these are the cells that are there. Water from this capillary oozes out or leaks out together with water soluble substances. Water and water soluble substance comes out, and these are then collected here in the spaces between these cells and these spaces. So it's collected, move out into the spaces between the cells of the tissue, and large proteins are remained here only. Large proteins and formed elements, these two are not removed. These two remain in that only, and this fluid which is released out is called interstitial fluid or tissue fluid, with the same mineral distribution, but the difference is that they don't have large proteins, they don't have formed elements. Now, understanding one more thing clearly is that when there is this capillary and you have tissues, so when liquid or water soluble substances oozes out and leaks into these spaces, this is called what? This is tissue fluid or interstitial fluid. However, if for example when this fluid actually goes into the lymphatic vessel, there's a whole system of vessels, lymphatic vessel, then this lymphatic, when it goes into lymphatic vessel, then this hole is called as lymph. So we'll talk about this also, but before that, this fluid helps in exchange of nutrients between blood and the cell, and blood and the cell exchanges always occur through this fluid. Okay, an elaborate network of vessel, which is called lymphatic system, collects this fluid and drains it back to the major vein. So what happens is that, first of all, that this is a capillary which oozes out or leaks out the water soluble substances, and they get collected in the spaces between these, and when they get collected, this is called tissue fluid or interstitial fluid. And when this fluid actually gets drained into lymphatic vessel, then this is called lymph, and this lymph is finally drained back to the veins, where it is called, of course, the blood, right? So this is the whole process. One more thing I want to tell you is that this tissue fluid or interstitial fluid that is there, this actually helps in exchange of nutrients and gases between the blood and the cell. So blood and the cell exchanges are done with the help of this. So this acts as a medium between blood and the tissue, right? Blood and the cells of tissue. Now we have lymph, which is a colorless fluid, of course, present in the lymphatic vessels. Fluid present in the lymphatic system is called lymph, and this lymph is a colorless fluid, like of course, like WBC. They are also colorless with their cells. However, this is the fluid which is colorless fluid, and they contain specialized lymphocytes. Lymphocytes are what? They're like WBCs, but they're not actually WBCs. Okay, why? Because lymph does not have formed elements, right? So because formed elements were not removed, large proteins and formed elements were not removed from the blood, right? They remained in the blood. But lymph has these specialized lymphocytes which also are responsible for immune responses. And if I talk about lymph, then it is also the carrier for nutrient, hormone, etc. Alright, and fat are absorbed through lymph in lacteal and present in intestinal villi, which means that intestinal villi, which are the finger-like projection in small intestine, these have lacteal, which is called lymphatic vessel, and lymphatic vessel consists of lymph, and this lymph absorb this fat, and the fat absorbed by the lymph in the lymphatic vessel is given back to the vein. So it is given back to the vein, and then from there vein gives it to the cells where they are absorbed. The fat is absorbed basically. And why it does not go directly into the blood vessel? The fat does not go directly in the blood vessel because the blood vessel does not have those larger pores. So fat cannot enter through these pores. These are very small. The blood vessel pores are very small, so fat cannot enter through that. So therefore it chooses another path. It first of all goes into the lymphatic vessel, and then from there it goes into veins and then into the bloodstream. So that's the whole way. Now we have circulatory pathways. Circulatory pathways are of like Patterns are of two types, which is open circulatory system and closed circulatory system. Open circulatory system is present in arthropods and mollusks, where heart of course pumps the blood and it passes through large veins. Remember this, large veins into open spaces or cavities called sinuses. And if we talk about annelids and chordates, they have closed circulatory system. What does that mean? Blood is of course pumped by heart only, but they pass through closed network of blood vessels. That's called closed circulatory system, and this pattern is considered to be more advantageous. Why? Because fluid flow is more precisely regulated. Alright, now all vertebrates have this muscular chambered heart. All vertebrates. When there is no exception, then you have to remember this, because they can directly ask and form this statement based question, or we would say that simple question of MCQ type, that they can say that, okay, tell me which of the following is present only in all vertebrates. Then you have to answer that as muscular chambered heart, all vertebrates without any exception. Now fishes have two chambered heart, atria and ventricle. Atrium and ventricle. Amphibians and reptiles except crocodile have three chambered heart, two atria and a ventricle. And crocodile, which is again a reptile, but it's an exception. Crocodile, birds and mammals have four chambered heart with two atria and two ventricle. Now they have given information about how single circulation takes place in fishes, double circulation takes place in amphibians, reptiles, we would say incomplete double circulation, and how double circulation takes place in there. Single circulation, complete double circulation, double circulation. Let's talk about it quickly. So we have fishes. Fishes basically, heart pumps deoxygenated blood, which is oxygenated by gills, and then this oxygenated blood is then supplied to body parts, and again deoxygenated blood is again carried from the body parts and is returned to the heart. So what happens is that there is single circulation only in case of fishes. So fishes first of all pumps the deoxygenated blood. So heart of the fishes pumps the deoxygenated blood to the gills, they oxygenate it and then gives it to the body. Body then supplies the deoxygenated blood again to the heart, because body is where carbon dioxide will be generated, and then this carbon dioxide makes it deoxygenated, and then deoxygenated blood is again supplied to the heart. So the blood only passes through heart once, so therefore it's called single circulation. Then in case of amphibians and reptiles, in amphibians and reptiles, we have left atrium receives oxygenated blood from gills, lungs, skin. Right atrium receives deoxygenated blood from other body parts, and they get mixed up in single ventricle, and then mixed blood is basically pumped, and this is called incomplete double circulation. So what happens is that oxygenated blood is obtained or oxygenated blood is received by left atria, and deoxygenated blood is received by right atria. Both of them are mixed in ventricle, and this mixed blood is then passed to the whole body parts like lungs and all those body parts. Again lungs oxygenates it and give oxygenated blood to left atria, and body again produces carbon dioxide, deoxygenates the blood and gives it to the right atria. Both of them are mixed up again and transported to the whole body parts like lungs and body parts. Now we have that's why it's called incomplete double circulation, and because of incomplete double circulation, mixing of blood, they are called cold-blooded organism. They cannot maintain their temperature. Body temperature actually changes with the surrounding. So but in mammals and birds, we have oxygenated and deoxygenated blood in left and right atria pumped to ventricles, their respective ventricles, without any mixing up, in separate circulatory pathways, and therefore this is called complete double circulation or simply double circulation. And therefore we are called as the birds and mammals are called a warm-blooded organism, because they can maintain their body temperature. Okay, they can maintain their body temperature. Alright, now you have to understand this very clearly, that in case of humans there is no mixing of blood, but in case of frog there is mixing of blood. Now we have human circulatory system. In human circulatory system, we have muscular chambered heart and network of close branching blood vessels and blood, and it's also called blood vascular system consisting of all these three. Understanding one point is that muscular chambered heart is present in all vertebrates without any exception, and understanding one more thing is that close branching blood vessels are present here because they are closed circulatory system organism, means the organism with closed circulatory system. And heart, if we talk about it first, then heart is a mesodermally derived organ which is situated in thoracic cavity between two lungs and slightly tilted to the left. It has size of clenched fist, and it is protected by a double walled membranous bag which is called pericardium. Pericardium is double walled, outer is called parietal pericardium and inner one is called visceral pericardium, and both of them encloses a fluid which is called pericardial fluid, and this pericardial fluid of course reduces tension and stuff. So pericardial fluid is present between those walls, double wall, right? So between those walls. Now our heart has four chambers, smaller upper chambers which are called atria and two larger chambers which are called ventricles. Now we have to understand that these two atria, if I talk about the small upper chamber to atria, are separated by thin muscular wall which is called interatrial septum, and lower to larger ventricles are separated by thick walled septum which is called interventricular septum. Then atria and ventricle of the same side are separated by thick fibrous tissue. You have to understand this is thick fibrous tissue is called atrioventricular septum, by septum which is called atrioventricular septum. And you have to understand one thing, that interatrial septum is thin muscular wall, interventricular septum is thick muscular wall, and in AV septum which is atrioventricular septum is thick fibrous tissue. However, the statement based question can be formed with this statement that however each of these septum are provided with an opening, and this opening is through which these two chambers of the same side are connected. So all these septum, whether it is interatrial septum, interventricular septum or atrioventricular septum, all of these have opening, and these openings are those openings which connect these two chambers of the same side together. Now opening between right atrium and right ventricle is guarded by valve. Valve are three basically muscular flaps or cusps, and if I talk about right atria and right ventricle, right atrium and right ventricle, then it's guarded by tricuspid valve which is made up of three muscular flaps or cusps, so it's called tricuspid valve. If I talk about left atria and left ventricle, then in that case it is guarded by a valve. Opening of between left atria and right ventricle is guarded by a valve which is called bicuspid or mitral valve. Then we have opening of right and left ventricle. Okay, if I talk about only interventricular septum, that has an opening which connects these two together, and they have opening, and this opening is guarded by a valve which is called semilunar valve. So you can see that they are guarded by semilunar valves. So basically left ventricle has this aorta, left ventricle is connected to aorta, and right ventricle is connected to pulmonary artery, and they are guarded by valve. Alright, now let me actually explain you with the help of this diagram that what is guarded with a valve and all those things. So basically you have this right atria, you have a right ventricle, both of them have opening, and this opening is guarded by tricuspid valve. You have left atrium and left ventricle, they have opening which is guarded by bicuspid valve. You also have right ventricle connected to this pulmonary artery, and there is an opening between them which is guarded by semilunar valve. And you also have left ventricle has an opening, there you have an opening, and this is also guarded by semilunar valve. It opens into aorta. So I hope you understood. And one more thing, that we have this atria, right and left atria separated by interatrial septum, and we have right and left ventricle separated by interventricular septum. So this is interventricular septum, and you have this as interatrial septum. And if I talk about one atria separated by another ventricle on its same side, that is by atrioventricular septum. Again, this atria, which is left atria, and this is left ventricle, they are separated by atrioventricular septum. I hope it made sense. Now, what are valves in heart? Valves in heart allows a flow of blood only in one direction, which is from atria to ventricle and from ventricle to pulmonary artery or aorta, but not the other way around. So they prevent the backflow of the blood. So valve allows the blood flow from only atria to ventricle, in both these, and from the right ventricle to pulmonary artery, and from the left ventricle to aorta. Alright, this is aorta. Now let's talk about nodal tissue. Nodal tissue is basically the specialized cardiac musculature. We know that heart is made up of cardiac muscles, and we also know the walls of ventricle are thicker, but the point to be remembered here is that specialized cardiac musculature is called nodal tissue in case of heart. They are distributed in the heart. First thing first, that patch of this tissue is present in the upper right corner of the right atrium. This is called sinoatrial node, and the mass of this tissue present in the lower left corner of the right atrium close to the AV septum is called atrioventricular node, which means that if I form a chamber like this, 4 chambers, then on the upper right corner of the right atrium, this is the right atrium, and on the upper right corner we have SA node. And if I talk about the lower left corner, we have mass of this tissue which is called AV node, and this AV node is near to the AV septum. You have to understand it's close to the AV septum. Now we have bundle of nodal fibers, atrioventricular bundle, continuing from AV node and passes through AV septum and emerges on the top of interventricular septum, immediately divides into right and left branch, and these branches then give rise to minute fibers throughout the ventricular musculature to the respective side, and these are called Purkinje fibers. What does it mean? Let me try to explain with the help of this diagram. So we have on the upper right corner sinoatrial node, on the lower left corner atrioventricular node. Now this is very close to this atrioventricular septum. Now what happens is that this whole thing is called atrioventricular bundle, AV bundle, and AV bundle also called bundle of His. Now this AV bundle continues from this AV node and passes through this whole thing which is called atrioventricular septum, passes through whole atrioventricular septum, then emerges on the top of this. What is this? This is interventricular septum, emerges on the top of interventricular septum, and then it immediately divides. It immediately divides into right and left branch, and these branches then produce fibers like this, and it covers the whole ventricular musculature, produces fiber and covers the whole musculature. Now these fibers are called Purkinje fiber. So what I'm trying to explain is the whole statement of NCERT. So you have to understand one by one how things are taking place, and remember that this sinoatrial node, this atrioventricular node is near to this atrioventricular septum. And one more thing, that this immediately divides here, where interventricular septum. What is this? Bundle of His, also called AV node. So remember these points. Now one more thing special about nodal musculature is that they have ability to generate action potential. Action potential, what are they? They are impulses. So they can generate impulses on their own without any stimulus by a neural system. So they are auto-excitable. Without any external stimuli, they can generate impulses, auto-excitable. But the number of action potential generated in a minute vary from different part to part, right, of this nodal musculature of the heart. Like for example, SA node can generate maximum number of action potentials maximum SA node can generate, and that is about 70 to 75 action potentials per minute. And SI node basically is also called pacemaker. Why? Because it is responsible for initiating and maintaining the contractile activity, rhythmic contractile activity of the heart. So it initiates, therefore it makes the pace, and therefore it's called pacemaker. Our heart normally beats 70 to 75 times in a minute, on an average 72 beats per minute. Now if I talk about cardiac system, cardiac cycle, then in that case we have four chambers of the heart. Now first of all, we are going to start from joint diastole. So all four chambers are in relaxed state in joint diastole. And in joint diastole, tricuspid and bicuspid valve are open. Blood from the pulmonary vein and vena cava flows into left and right ventricle through left and right atria, and semilunar valves are closed at this stage. So this is the whole joint diastole description. So what they are saying is that during joint diastole, which is about 0.4, the total cardiac cycle is about 0.8. So in joint diastole, which is about 0.4 seconds in one cardiac cycle, what happens is that basically the blood, tricuspid bicuspid valve are open, so blood basically flows from vena cava and pulmonary vein to right atrium and left atrium respectively, and then they get transported to right ventricle and left ventricle. Tricuspid valve are open, semilunar valve here and here are closed. That's the whole thing here. Now we have action potential generated by SA node. SA node generate action potential. Because of that, both atria contracts, which is they cause simultaneous contraction, atrial systole. All the things are same, but what the difference is that flow of blood into the ventricle is increased by 30%. Flow of ventricle is increased by 30%, which means that blood is given by the body through vena cava and by the lung through pulmonary vein into right and left atria, then right and left ventricle. Tricuspid and bicuspid valve are open, semilunar valve are closed. However, this blood flow has been increased by 30%. Now what will happen is that, and this action potential was generated by SA node, which is present in upper right corner of the right atrium, right? Action potential generated. Now this action potential is conducted to ventricular side. How? The conducted ventricular side by AV node and AV bundle. So the AV node, which is present on the lower left corner of the right atrium, and the AV bundle, which passes the whole AV septum. So basically what happens is that they conduct this action potential to the ventricle side, and then finally bundle of His then divides, and then Purkinje 5 transmits it through the whole ventricular musculature. And this causes what? This action potential causes ventricular systole. Alright, so this action potential causes ventricular systole. Now atria is relaxing, and that relaxation of atrial diastole is coinciding with the ventricular systole. So what is happening is that atria is relaxing, but ventricular systole. So first of all, joint diastole took place, then we have atrial systole, and then we have ventricular systole, and there we also have atrial diastole taking place. And then ventricular systole is happening right now. This whole thing is happening together with atrial diastole. Now what happens is that when ventricular systole increases, mean the pressure increases, then it causes closure of first of all tricuspid bicuspid valve. First thing first happens is the lub sound produced because of this closure of tricuspid bicuspid valve. Why tricuspid bicuspid valves are closed? Because if the pressure inside right ventricle and left ventricle are increased, then they can cause backflow, right? But backflow is prevented by the valves, and because of that, these valves has to be closed. So they prevent backflow, right? So they close them, and because of that, lub sound, first sound is produced at the end of ventricular systole. But one more thing happens is that because more pressure, more and more pressure increases, so because of that, semilunar valves are pushed open. Semilunar valves guiding pulmonary artery and aorta are pushed open and forced open. So because of that, they start moving into the circulatory pathways. Now because of that, so basically when the pressure is increasing first, so because of that, tricuspid and bicuspid valves are closed, semilunar valves are open, and now blood starts flowing into this system and this system, these two circulatory pathways. Now ventricular now relax, ventricular diastole, and the pressure falls. Now ventricle start relaxing, this diastole, joint diastole starts coming, where both of these, atria is relaxing, ventricle is relaxing, joint diastole. And this joint diastole is when pressure inside the ventricle decreases. And when pressure falls, first thing first, there is closure semilunar valve to prevent the backflow. And then ventricular pressure or declines further. Because of that, tricuspid bicuspid valve pushed open. How? By the pressure of atria exerted by the blood which was being emptied into them by the veins. So what happens? Let me explain you. Basically, ventricular pressure falls, so they are just diastoling. These two are diastoling. Ventricular pressure falls, and because pressure falls, first thing first is that semilunar valves are closed. So we have the closure of semilunar valve, but further declines pressure inside the ventricle, so tricuspid valve and bicuspid valves are forced open by the blood pouring out by different veins, pulmonary vein and this vena cava vein into them. So blood is being poured into them. Because of the force, tricuspid and bicuspid valve are pushed open, and this is where closer semilunar valve cause this dub sound at the end of this joint diastole. Alright, now we know that how dub and lub sound are produced. Moving on, we have blood moves freely again to the ventricles like this in joint diastole, and then again SA node generate action potential which cause the systole of atria, and then 30% increase blood flow is increased into the ventricles, and after that ventricles also contract, and when ventricle contract, tricuspid valve is closed, bicuspid valve is closed, which produces the lub sound first, and this lub sound is produced, and because further increases more pressure, further increases more, so semilunar valves are pushed open, and when semilunar valves are pushed open, the blood starts flowing into the circulatory system like this and this. And then what happens afterwards? The pressure declines, and pressure first of all declines. Then first thing first is that semilunar valves are closed, which produces dub sound, and then when it declines further, then tricuspid bicuspid valves are pushed open, so atria and ventricle again pushes the blood to go into the ventricle, and this is called joint diastole. And after that again SA node basically generates the action potential, and SA node generate the action potential, then in that case they causes these two atria to again contract and causes atrial systole, then it will cause ventral systole, and this whole cycle sequence of process will continue again and again and again. So sequence of events in the heart which is cyclically repeated, this one cycle when it's repeated again and again, this one cycle is called cardiac cycle. It consists of systole and diastole of both atria and ventricles, right? Heart beats 72 times per minute. Alright, so basically 72 time beats takes place in a minute, which is 60 seconds, so one beat will take 0.8 seconds. That's why we were saying the total cardiac cycle takes 0.8 second to occur. Now we know that in each cardiac cycle, each ventricle pumps 70 ml of blood, so that's called stroke volume. So stroke volume is blood pumped by ventricle in each cycle, which means in each beat. But when this stroke volume is multiplied with heart rate, then it gives cardiac output, which means cardiac output is the volume of blood pumped by each ventricle per minute, which means in 72 beats, which is 5000 ml or 5 liters in healthy individual. And we also know that the body has ability to alter stroke volume. It can change stroke volume and heart rate also. So that is done by cardiac output. Sorry, it can also change cardiac output, and that we can see this can be done in case of athletes also. We can see in athletes they have more cardiac output than ordinary man, right? Higher cardiac output. Now one more thing, that there are two prominent sounds that are produced, as I told you. First heard sound is lub sound, and that is caused due to tricuspid bicuspid valve closure, and second heard sound is dub sound due to closure of semilunar valve. Remember my word, opening of valves, opening of anything does not cause any sound. Only closure of these valves cause sound, as I told you, explained very well. Now we have electrocardiogram. You also have seen, like you all have must have seen the scenes from hospitals which show P P P P, and when P sound is produced, this means that person goes into a cardiac arrest. What is cardiac arrest? It is the condition where nodal tissue does not work, and when nodal tissue does not work, then there can be no impulse generated. No impulse is generated as a node or AV node, whatever. They cannot generate the impulse, and because they cannot generate impulse, the heart cannot beat. And once heart stops beating, the person goes into this condition, and of course the person cannot survive, right? Machine is called electrocardiograph. However, the graphical representation which is there is called electrocardiogram. So you have to remember the difference. Graphical representation actually is called gram, and if I talk about the machine, that's called the graph. And what is the representation of it? It's the representation of electrical activity of heart during a cycle. Okay, so we have standard ECG. In standard ECG, patient is connected to 3 leads, and 2 leads are connected to each wrist, and 1 lead is connected to left ankle to monitor the heart activity. For detailed evaluation of heart's function, multiple leads are attached to the chest region. So this can be asked for statement based question. Remember, all these statements which I highlighted with this color can be asked in statement based type questions. Each peak in the ECG, if you talk about the standard ECG, then each peak in case of ECG is identified with letter P to T, and then they represent basically specific electrical activity of the heart. First thing first is that P wave will represent electrical excitation or depolarization of atria. Depolarization means that impulse is conducted, impulse is fired in that region, so that's called depolarization. So if impulse is fired for atria, then it's called that atria are depolarized. So P wave represents depolarization of atria, and this depolarization will cause ultimately the contraction of atria, and this is done by SA node initially, right? Now QRS complex is another peak which represents depolarization of ventricle. Depolarization of ventricle causes ventricular systole. So after just Q, we can understand that the beginning of systole, ventricular systole has taken place. One more thing, that we also know that atria will diastole, and atria will diastole, that should be shown here, but that's not shown here, right, in any of the peaks. So I would like to tell you one thing, that atria diastole basically is generated. However, this is suppressed. Atrial diastole is suppressed by the ventricular systole. So this QRS complex is so high and so peaked. You know that ventricle is very thick and large muscular. So we all know that ventricle will suppress any excitation or any electrical activity shown by atria because it's very small, and because of that it's suppressed, and therefore it's not seen here. However, we know that NCERT has also written this, that ventricular atrial systole and atrial diastole and ventricular systole takes place simultaneously. I'll show you that line. There's written this, that atrial systole and ventricular systole and atrial diastole takes place simultaneously, and that's why we cannot see the atrial diastole a peak, right, because it is suppressed here. So this is ventricular systole plus atrial diastole. Alright, then we have T wave. T wave represents return of ventricle from excited to normal state, which is called repolarization, and end of T wave means that there is end of systole. Now there will be like relaxed state of all four. So when all 4 are relaxed in joint diastole, there is no electrical activity, and when there is no electrical activity, the heart remains silent, and there is no peak. Alright, so there is no peak. That's what I wanted to tell, that this is the condition for joint diastole. This is where atrial systole takes place, this is where ventricular systole takes place, and this is diastole of ventricle, and this is the joint diastole. And you have to understand, when it is systoling, then it's systoling atria, otherwise all the time it's diastoling only. These are the other things. I mean, if it is systoling here only, ventricle systoling here only, then all other time it will diastole. In case of ventricle systole also it will diastole. Alright, made sense, I think. Now by counting the number of QRS complexes, it's like peak, so it's very visible in the whole gram, which is the graphical representation. So we can say that by counting that in given time, heartbeat can be calculated, and different ECG obtained from different person roughly have same shape. So if there is any deviation, then we can say that there will be abnormality or disease. One thing I would like to highlight is that for given lid configuration, we would have same individual ECGs. Just wanted to highlight. Now we have double circulation. In double circulation, as I told you, this is in birds and mammals, and also one more crocodile, which is a reptile. Crocodile, which is a reptile, but double circulation is present in there also. If I talk about double circulation, then this is called complete double circulation. Blood flows strictly by fixed route, okay, which is through blood vessels. And let me talk about blood vessels. Each atria and veins are blood vessels. They have three layers. Inner layer is made of squamous endothelium. Endothelium is basically epithelium of blood vessel. So inner layer is squamous, and that's called tunica intima. Middle layer is made of smooth and elastic fibers, and that's called tunica media. And the external layer is made up of fibrous connective tissue with collagen fibers, that is called tunica externa. So these two questions are asked definitely in exams, that tunica media is made up of what? So smooth muscle and elastic fibers, and tunica externa is made up of fibrous connective tissue with collagen fibers. So you can see media is made up of elastic fibers, and externa is made up of collagen fibers, and this is smooth muscles. Now, as mentioned earlier, blood pumped by right ventricle enters the pulmonary artery, whereas left ventricle pumps blood into the aorta. So what happens is that deoxygenated blood pumped into pulmonary artery is passed on to lungs. Alright, deoxygenated blood is passed on to lungs, whereas oxygenated blood is carried by pulmonary veins into left atrium. This pathway constitutes pulmonary circulation. So this is about pulmonary circulation, and oxygenated blood enters the aorta through network of arteries, capillaries, arterioles to the tissue, and then from where deoxygenated blood is carried by venules, vena cava to the left atrium, and then it's pumped again. So this is called systemic circulation, which takes place between tissue and the heart, and it takes place between lungs and the heart. So these are pulmonary and systemic circulations. Systemic circulation provides nutrient, oxygen, essential substances to the tissues, and takes carbon dioxide, harmful substance, for elimination. But one more point to be remembered here, there is unique vascular connection between digestive tract and liver tract. So digestive tract and liver tract are connected together with hepatic portal system. Hepatic portal vein is basically a vein which carries blood from intestine to liver before it gives it to systemic circulation. So first of all, intestine gives it to liver, and then gives it to the systemic circulation back. So intestine directly does not give it to systemic circulation. That's why it's called special unique connection, vascular connection between intestine and the liver. And there's also coronary blood vessel system, which is exclusively for circulation of blood to and fro from cardiac musculature. Let me explain you pulmonary and systemic circulation with the help of this diagram. So first of all, what happens is that pulmonary vein gives the blood to the, whatever is vein will give the blood to the heart, and whatever is the artery will take away the blood away from the heart. So what happens is that pulmonary vein gives blood to the left atria, and basically then right ventricle gives blood back to the lungs through pulmonary artery, and blood is collected from vena cava to right atria, and then left ventricle gives oxygenated blood back to the body parts again. So this is called systemic circulation. Now one more point to remember is that regulation of heart activity we have to understand, but we know this heart is auto-regulated. Normal activities of the heart are regulated intrinsically by nodal tissue. So therefore it's called myogenic, because muscle controls it, okay? It does not depend on signals from nervous system, neural system. It can regulate itself intrinsically by nodal tissue. Therefore it's called auto-regulated and myogenic. But there is special neural center in medulla oblongata which moderates the cardiac function through ANS, autonomic nervous system. So there is medulla oblongata, where there is neural center, special neural center, so and that can moderate cardiac function through ANS, autonomic nervous system. And like autonomic nervous system has two types: systemic nervous system and sympathetic nervous system and parasympathetic nervous system. Sympathetic increases the heart rate, heartbeat, contraction strength, and cardiac output. However, parasympathetic decreases the heart rate or brings it to the normal level. Not decrease in a sense that it decreases, but it brings it to the normal level, the heart rate, speed of conduction, reaction potential, and thereby cardiac output. One more important thing is that hormonal system also controls the rate of heartbeat, which is adrenal medullary hormones can also increase the heart cardiac output. You have to understand, adrenal medulla is responsible for increasing the cardiac output, not the adrenal cortex. Just remember, this can be asked very easily. Now we have disorders related to circulatory system. Now in case of circulatory system, we have high blood pressure, which is hypertension. Hypertension is a term for blood pressure which is higher than normal. So if repeated checks shows that the blood pressure of individual is about 140 to 90, right, 140 over 90 or higher even, then the person is hypertension, means the disease which person is suffering from is hypertension. And you have to understand is that normal systemic or pumping pressure is 120 and the normal diastolic resting pressure is 80 mmHg, or else a person suffering from hypertension. Now we have hypertension, then we would say that high blood pressure, hypertension can lead diseases to heart disease, right, can lead diseases of heart also, and can also affect brain and kidney, right. And then we have coronary artery disease, which is also called CAD. It also is referred to as atherosclerosis, however you pronounce, I don't know, but it affects the vessels that supply the blood to the heart muscle. So the vessels which are called coronary, that it mentioned, NCERT mentioned that there is a coronary system of blood vessels for exclusively for heart. So that blood vessel supply blood to the heart. Now that vessel is affected in this disease. How is it affected? Basically, there are deposits of calcium, fat, cholesterol, or fibrous tissue in the vessels that affects the vessel actually, because that will block the vessel. These depositions will block the blood flow, and they will block the blood flow. They'll make the arteries narrower, and they will affect the vessel, and that's the disease about, right. And once, of course, think about it, if vessels are affected, then blood supply is not proper to the heart, and if blood is not supplied properly, oxygen is not supplied properly. And if, for example, there's a time when the whole blood cannot be supplied, there's big chunk of this deposit here, and then whole blood is not supplied to the region of heart, that region will be declared as dead. And we would say that if that's small, then it's minor heart attack. If that's big, then it's called major heart attack. And there will be acute chest pain. So angina, which is angina pectoris, is symptom of acute chest pain. So there is symptom which is called acute chest pain, where no enough oxygen is reaching the heart muscle, because of, again, the same, it occurs due to the condition that affects the blood flow. So the condition which affects the blood flow is CAD, because it affects the blood vessel. So it affects the blood, the condition which is CAD, it affects the blood flow, and because of that there is a symptom which is called acute chest pain, because of course blood is not reaching, oxygen is not reaching the heart muscle. So because of that there is acute chest pain, that's called angina pectoris. It can occur in men and women of any age. However, it's more common in middle-aged and elderly age women and men. Now, heart failure is the state of heart where heart is not pumping effectively. Means heart is pumping, but it's not pumping effectively to meet the needs. It's also called sometimes congestive heart failure, because congestion of lung is one of the major symptom, and it's the symptom, it's not the cause. Just mind it, because I had also assumed that it was a cause. No, congestion of lungs is a symptom of this heart failure, Means if for example person's heart is failing or it's not meeting the needs effectively, it's not pumping the blood to meet the needs of the body, demands of the body, then in that case one of the symptoms that person going through can be or is congestion of lungs. Now heart failure is not same as cardiac arrest, not same as heart attack. All three statements, all three words are different. Heart failure is when enough or effectively heart is not pumping the blood. Heart cardiac arrest is when nodal tissue is damaged and heart stops beating. As I mentioned, P sound is there in electrocardiogram, then there is like person nodal tissue is not working and then heart stops beating. Impulse cannot be generated. Heart attack is when the person muscle is suddenly damaged, and how the muscle can be damaged by inadequate blood supply. And how inadequate blood supply can be there when the blood vessel transporting the blood itself is damaged, and that happens in CAD, which is coronary artery disease. So that was the point I wanted to mention. Now all these things you can read by your own, you all must have NCERT, but one thing which I want to clarify here is this: these statements are very important. Like spaces between the cell in the tissue contain fluid derived from the blood. So of course we have the capillary and we have the tissues. So water soluble substance, water passes through these and then they are collected here. Now the liquid or fluid which is collected here between the cells, this is called tissue fluid, and this tissue fluid has almost same blood composition as the blood. However, it does not have protein content, does not have formed elements, because they cannot ooze out, they cannot leak out of that capillary. All vertebrates, few invertebrates, but all vertebrates have closed circulatory system. This one more statement based question can be formed from here, that all vertebrates without any exception has closed circulatory system. All vertebrates without any exception has closed muscular heart, right? It was mentioned, no, that they have closed heart, not closed, muscular heart. Another thing is cardiac musculature is auto-excitable. SA node can produce, is called pacemaker, and can produce maximum number of action potential, which is 70 to 75 per minute, and it sets a pace for the activity of heart. So therefore it's called pacemaker. Electrical activity of heart can be recorded from body surface by using electrocardiograph, which is the machine, and the recording is called electrocardiogram, which is the graphical representation. So graphical representation is gram and machine is graph. Another statement is, though the heart is auto-excitable, its function can be moderated by neural and hormonal mechanism. Neural mechanism, which is located in medulla oblongata, through ANS, which is autonomic nervous system, it regulates. By sympathetic and parasympathetic, it increases the heart rate and it decreases the heart rate, right? And another is hormonal mechanism. Hormonal mechanism is basically the adrenal medulla. Adrenal medulla is basically the one which actually increases the heart rate. You have to understand this. I think I have covered it everything. So do let me in the comment section how you liked it or whatever your opinions, and thank you so much for watching. Bye.